We can get started, I think.
Yeah, we can start. Very good evening, everyone, and welcome all to again the monthly research article spotlight. And let me welcome Miss Harshita Nagar. She will present her research work in mechanochromic luminescence in copper nanocluster.
No, there is some echo. There is some echo. Please redo.
Can you
Echo. There is some echo.
anyone?
Please switch off multiple devices.
I'm audible now. Is it okay?
It is okay now.
Oh. Very good evening, everyone, and welcome all you again here to monthly research article spotlight. Today's talk is by Miss Harshita Nagar. She completed her bachelor's and master from Mahatma Gandhi Kashi Vishwanath University from Varanasi. Her topic will be mechanochromic luminescence in copper nanocluster, resolving structure transitions through microcrystal electron diffraction. So over to you, Harshita. Please maintain time for the 30 minutes and 20 minutes for the discussion. So stage is yours.
Thank you, Deepak Bhaiya, for the introduction. My slides are visible.
Coming. It is coming. Yes, it has come. Make it full screen though. Yes, good. It is good. Go ahead.
Good evening, everyone. Today I'll be presenting my work, which we have published recently in ChemCom, titled as Mechanochromic Luminescence in Copper Nanoclusters, Resolving Structural Transitions Through Microcrystal Electron Diffraction. It was published on 11th June 2025. So coming to the introduction of nanoclusters, we all are aware atomically precise metal nanoclusters are ultra-small particles of size 1 to 3 nanometer, and they lie in between atomic regime and plasmonic nanoparticle regime. These nanoclusters are stabilized by ligands, and these ligand controls the shape, size, and nuclearity of these nanoclusters. So there are various characterization techniques to characterize these clusters, like UV visible spectroscopy, mass spectrometry, for structure determination, single crystal XRD, SEM, TEM, and many more. But the crucial aspect is to determine the crystal structure of these nanocluster. So the central aspect of any material is a structure, because these nanoclusters have a wide variety of application in the field of energy storage, catalysis, sensing, and all these applications are associated with their physical and chemical properties, which are governed by the crystal structures of these nanoclusters. So there are multiple ways for the structure determination, like XRD, single crystal X-ray diffraction, electron diffraction, neutron diffraction, but my work is based on microcrystal electron diffraction. So before going further, we all are aware about single crystal X-ray crystallography, because it is found to be the gold standard for the structure determination. But if we will talk about X-ray, these are carried out with the typically used wavelength of around 1 angstrom. And in X-ray crystallography, large crystals are needed. If you have a protein samples or something, like in that case, it is very time consuming to grow large size crystal as well as difficult. So there is a need in crystallography to develop methods for micro and nano size crystals. Then there we use microcrystal electron diffraction. So it is a cryo-electron microscopic techniques which is used to determine the structure of nano and microcrystal, and microcrystal electron diffraction combines TEM, that is transmission electron microscopy, with electron diffraction. So the advantages I already told. Since X-rays have a wavelength of around 1 angstrom, electrons have very shorter wavelength of around 0.025 angstrom at 200 keV. And there is a very strong interaction between the electrons with the matter, because of which even the crystals of few nanometers can be studied using this technique. So microED enables structural analysis of samples that are too small for traditional single crystal X-ray diffraction technique. So this is the schematic of micro-ED data collection, where we take the where the sample is kept or the crystal is kept on the stage and the electron beam incident on it. Then there is a continuous rotation of the stage, and then we collect multiple diffraction data using a high-speed detector. And then these diffraction data collectively will combine together to give a data set, which we use to process further to get the structure. And usually, when this electron beam falls on these crystal, it occurs in the presence of a very low electron dose rate, like let's say 0.01 electron per angstrom square per second. Then coming to the difference between the microcrystal electron diffraction and single crystal X-ray diffraction, as I've mentioned earlier, first difference is the size difference. In case of X-ray diffraction, the crystals of few microns of size can be used, but for electron diffraction, even smaller size can be used. And the difference is due to since the wavelength of the electron is around 0.025 angstrom at this particular 200 kV voltage, and for X-rays, the wavelength is 1.54. So the major difference is how their diffraction patterns appear. For X-ray diffraction, if you see, the diffraction appears found to be something like a circular or spherical, but in case of electron diffraction, they appear to be linear. Now the difference lies is this diffraction pattern arises when there is when the surface of the evolved sphere lies on the surface of these points of these reciprocal space, these reciprocal points. So in case of electron diffraction, where wavelength is very small, there is a relation. Radius of the evolved sphere is inversely proportional to the wavelength. So in case of electron diffraction, where the wavelength is very small, the radius of evolved sphere is very large. So if we will consider a particular reciprocal space, then it will appear as a plane or as a linear. That is why the major difference between the diffraction pattern in both the techniques is because of their wavelength. Now coming to the history of micro-ED. Micro-ED was first developed by Tamer Gunen Group in Genelia Research Campus in 2013, and with the students with the student Brent Nanega, who is our collaborator also, where they first started with the protein sample, where again they have used a very low dose rate for the structure determination of the protein. So they started their work with the lysozyme protein in 2013. They tried to solve the structure of lysozyme with the resolution of around 2.9 angstrom. Then over time, they improved the resolution to 1.5 angstrom using different softwares, different techniques, and good camera also. So, and then they extended the application of this micro-ED towards many other fields like metal organic frameworks and others. Now, the crucial step is the sample preparation technique in case of cryo-EM. So there are multiple ways in which we can prepare the sample. Like if you can see the first one, just take the crystals, load it into the TM grid and collect the data. Other way, if the crystals are already in the vial inside the solvent, we can just use the pipette to make them small and then dropcast it onto the glass into the TM grid. Or other way, you can just grind it on the mortar pestle or crush them between the glass slides. So there are multiple ways. We mostly use this type of technique where we take the crystalline powder and load it onto the TEM grid. So if I'll consider my work, what I have used is, we took the crystal, just crushed them between two glass slides, then loaded it onto the TEM grid, and then this TEM grid is clipped between C-clip ring and C-clip. And then this clipped TEM grid is kept into the autoloader, which we kept inside the cryo-EM to collect the data, to collect the further data. And after that data collection, we get the structure. So this is the overall micro-ED workflow. It is having different type of presets, 4 different type of presets. First is the Atlas view or Atlas preset, which will give you the whole magnification, like global map, complete view of the grid. Then after that, we will select one of the particular grid square, which is known as a grid square preset, which will give the entire view of one grid square. After that, we will select one particular crystal that is known as the search or autocentric preset, where we will just check the crystal of our interest. And then last is the diffraction preset, where we will diffract that particular crystal for collecting the electron diffraction data. Now coming to the instrumentation part, we have done this work using a Krios G4 cryo-EM from our lab, where the source is XFEG. Dose rate we have used is around 0.01 electron per angstrom square per second with the exposure time of 1 second. And detector we are using here is Theta D CMOS camera. And about this angle rotation, I will explain you further. Now coming to the work which we have done in this work. So we started with the 3 copper nanoclusters with 4 nuclearity. All these nanoclusters are protected with different type of ligands. These nanoclusters are synthesized and well characterized in our lab from our seniors, and their structures are also determined. But since it was our first work of electron diffraction, we started with the small nuclearity cluster. So these are the UV visible spectra of all these 3 nanocluster. First I'll explain. This one is the copper 4 MNA2 DPPE2. MNA is mercapto nicotinic acid. DPP is diphenyl phosphinoethane. So for this nanocluster, it is having a peak at 297 and 380. Then coming to other 2 nanoclusters, which is copper 4 protected with orthocarborane 9 thiol ligand, and other one is copper 4 which is protected with metacarborane 9 thiol ligand, and these are the UV visible features of these clusters. And these are the corresponding mass spectrum of this, from where we got these compositions. Now in this work, we took all these 3 clusters for different studies. So we started our investigation with the first cluster, that is copper 4 protected with mercaptonicotinic acid and diphenyl phosphinoethane. So for this, we started with this clusters to see the reliability of micro-ED for the accurate structure determination of these nanocluster, like to validate the micro-ED result with that of the single crystal XRD. So these are the TEM images of the microcrystals of this nanocluster. These were present in a DMF, so we just pipetted it and we just broadcast it onto the TEM grid. So these are the TM images of those microcrystals. And as I've mentioned, this is a continuous rotation electron diffraction. That means sample is kept on the stage and stage is rotating, and then we collect multiple diffraction data. So here we used the angle from minus 50 degree to plus 50 degree, and we rotated it every time with 1 degree tilt per image. And this is the initial data. This is the initial diffraction frame which we got for this cluster, and we were able to get the resolution of around 0.8 angstrom. And this is the micro-ED structure of copper 4 MNA2 DPPE2 nanocluster, which has a copper 4 S2 core, which is made up of the 2 copper 2 nanoclusters, and where sulfur is acting as a bridge. It is having 2 types of copper atoms. One is pentacoordinated, which is connected with phosphorus, nitrogen, 2 types of sulfur and 1 copper. Other one is tetracoordinated, which is connected to oxygen, phosphorus, 1 copper and 1 sulfur. So this is the overall micro-ED structure which we got from here. And this is the 3x3x packing along the B axis. So from here, we got the idea of how to use this technique to determine the structure of these nanoclusters. And as I've mentioned that we have to rotate the stage from various angles. So this is the TEM tilt series images of this copper 4 MNA2 nanoclusters captured at different tilt angle from minus 50 degree to plus 50 degree by rotation of 5 degree every time. So this was the first example which we took. Now we compared the results of the micro-ED which we got, like the unit cell parameters and space group, with that which was already reported from single crystal XRD, and they were found to be very much similar. So that from this we can see the reliability of the micro-ED technique for our type of system. Now coming to the second cluster which we used was copper 4 nanocluster, which was protected with metacarborane 9 thiol ligand. So here micro-ED give us the new insight about the solvent inclusion within the molecule. So in case of this copper 4 M9CBT, these are the crystals which we have chosen, and in all these crystals we can see the size is even less than 1 micron. And again, here again, we rotated the stage from -40° to +40° with 1 degree tilt per image. And in this case, we were able to get the resolution of around 0.9 angstrom. But the major insight which we got was the presence of the toluene solvent within the unit cell of the nanocluster. This was the new insight which we got from microcrystal electron diffraction, which we were not, which we didn't observe earlier from single crystal XRD. And this shows the crystal packing along 3x3x3 axis. And now the question is why there is like how we can see the presence of toluene here. So there is a very strong interaction between the electron and matter. This could be the reason that it is more sensitive technique, and that's why we are able to see such type of solvent inclusion within the cluster. And since there was a solvent inclusion within the unit cell parameters and space group, everything varied from single crystal XRD. So from here, from the first, we consider the reliability of microED. From the second example, we got the insight of the solvent inclusion within the molecule. Now coming to the third work, before that, many copper nanoclusters show mechanochromic luminescence. And from our group, from Arijit Bhaiya's paper, he also showed copper for OCPT and copper for MCBT shows mechanochromic luminescence, as well as some other groups also showed copper iodide clusters also show mechanochromic luminescence. But the challenge is how grinding induced structural changes correlate with the altered luminescence property, leaving fundamental question about structural integrity and solvent interactions unresolved. So basically, in all these work, they have shown that they grind the crystals. Before grinding, they have some emission. After grinding, their emission change, and upon solvent exposure, their emission get reversed. But the structure, but the phenomena, this mechanochromic phenomena was unexplored earlier, which we tried to explore here. So we used microED here for the investigation of the mechanochromic phenomena in copper for OCBT cluster by providing direct structural evidence of this amorphization recrystallization technique. If you can see here, firstly, these crystals before grinding, they show green emission. And then after grinding for a few minutes, their emission changed to yellow color. And then upon solvent exposure, their emission is coming back. So we used microED in all these three stages. So at stage 1, before grinding, we did the microED, and this is the diffraction pattern which we got. Again, the resolution was around 0.8 angstrom, and the crystal size which we used here was even less than 1 microns. And the crystal, and this is the microED structure which we got from such tiny crystals. After that, since we grinded it for a time, and there is a change in the emission from green to yellow. So it was reported that it become amorphous. So it become amorphous because there is a disruption of the long range order here. So if you can see, there is a basic difference. Like if your pattern is if you have a crystal, you will get regular diffraction spots. But if the system is amorphous or it is not crystalline, we will not see these diffraction spots. So here this spots disappear. So there is no defined structure from this. And then upon solvent exposure, we are getting back the diffraction pattern, which shows that the emission is back. And now if you will see the size of the crystal, earlier it was in the in the scale of 2 microns, but now after grinding and solvent exposure, the crystal size is very tiny. It is even less than 500 nanometer. So our objective was to get the crystal structure of this cluster using this tiny crystals. So we used this tiny crystals to get the complete structure of this copper for OCBT. So from here, what we are observing that this change in emission, and again getting the change in emission upon grinding, and then getting amorphization, and then recrystallizing it upon solvent exposure. This shows that there is a disruption of the long range order, but there is but the chemical structure is still same. So it shows that there is a loss of long range order, but there is no change in the but there is no chemical changes. So from here, we were able to get the crystal structure of copper for OCBT from nanometer size crystals. Then we compared the single crystal XRD results with two of the data. That is the first one, the micro-ED structure of this one, that is before grinding, and the micro-ED structure after solvent vapor exposure. And in all the cases, we are able to get the similar unit cell and similar space group. So the conclusion of my work is, first, with our first example, validation of micro-ED accuracy. Micro-ED demonstrated as a reliable and rapid technique for precise structure determination of copper nanoclusters, and we were able to get a good atomic resolution of 0.8 angstrom for copper 4 MNA2 nanoclusters, and for other two also, we were able to get around 0.8 to 0.9 angstrom resolution. From the second example, micro-ED identified a toluene solvent molecule within the unit cell, which was missed in single crystal XRD. And third, direct visualization of mechanochromic mechanism. That is, for this copper 4, micro-ED provided direct structural evidence of the luminescence mechanism, which arises from the disruption of the long range order, followed by recrystallization, rather than any chemical deformation. And then we compared the structure before grinding and after solvent recovery, which confirmed the same structure throughout the mechanochromic cycle. And coming to the future direction, we started with this very small nuclearity cluster of copper 4. Now our aim is to develop this technique for the large nuclearity clusters as well. So we are working with the AG29 nanocluster, which is protected with benzene dithiol and triphenyl phosphine. In this work, what we have found it in this particular cluster, it is having two polymorphs. One is cubic and one is trigonal. So here in a particular grid square, as I've mentioned, we have a grid square preset. So in a particular grid square, we are able to get both the polymorphs. Now we are trying to solve their crystal structure. And at last, I would like to thank Sir for giving me this opportunity to learn this technique. Our collaborators, Professor Brent Nenega from Arizona State University and Dr. Thomas Basse for sending us the carborane ligands, my funding agency UGC, IIT Madras, Department of Chemistry, and my lab mates. Thank you.
Thank you so much, Harshita, for a very nice and interesting presentation. Now, floor is open for the discussion. If you have any question, please ask.
So, Harshita, excellent talk. So, I am curious to know about the micro-ED data. So, in case of your MNA2 DPP2 data, the fringe lines are discontinuous, what I can see there. But in case of M9 CBT ligand, the data is somewhat continuous. So,
can you repeat, Sombhaiya?
you can see in figure B, there the fringe lines are there. So those
You are
lines
talking
are,
about diffraction pattern?
yes, exactly. So I want to get a brief idea about what are these fringe lines and what are these concentric circles. What does it denote? Yeah, can you
These
briefly tell that?
concentric circles are just to represent the resolution range. They are not these are the manually these just show the resolution range. This is not to do anything with the diffraction pattern.
Okay.
Just
And what about
like
this
suppose if you will see here, till here it will show the resolution of around 2.49, and if your diffraction spot lies here, it will show the range. So this is just to show the range.
Okay. And what about these fringe lines? Because here it is discontinuous, but if you go to your MCPT spectrum, it is more continuous.
These are not the fringe lines, these are the diffraction pattern, and it also depend upon the unit cell parameters.
Okay.
So since in case of copper 4 M9 and copper 4 O9, both are having the carborane ligands, but that copper 4 MNA DPP, the ligand is different. So the unit cell parameters are also different. So the diffraction pattern, these represents the HKL planes.
Yes.
So that is why it depends on the what are the unit cell parameters which we are getting.
You can tell that this is different planes. The diffraction
Yes.
pattern is from different planes.
Different
So
planes,
if you
HKL.
get the same plane of that other structure, you can see this kind of diffraction.
And this is one of the frame. Like we have a set of data, like from -50 to +50, we will have 100 diffraction patterns. So each will look different.
Okay. And in your OCBT also, I can see that the these diffraction patterns are more scattered in case of 3 if you compare it to 1.
I think it could be because of the crystal size also, because if you will compare the crystal size from here to here, there is a huge difference.
Harshita, wait. That doesn't depend on the crystal size. The thing what Bijesh was telling, they are different planes.
Okay.
So in one of the plane, the figure, say, first and third, they might have some similar planes
Yes.
somewhere. But the picture what you have put is might be some first plane and the other one will be, say, 1/20th. So
Huh.
that will
Correct.
vary. So it doesn't
Yes.
depend on the size of the crystal. And each
Mhm.
frame is kind of fingerprint of that
Particular
whatever,
angle.
yeah, particular angle. So these things can change.
Yes. Because we have a set of data,
Yeah.
diffraction data.
Okay, thank you.
Harshita,
Mm-hmm.
very nice talk. Like, I'm just curious to know that during the sample preparation of your micro ED, you are grinding the crystals, right?
No,
But
crushing just.
yeah, just crushing. But you
Yes.
are applying some mechanical force or energy. So
Yes.
we know that our clusters undergo, or any crystal can undergo phase transition, or it can introduce defects in the crystal lattice, or some polymorphism can happen under mechanical energy.
Mhm.
When you are doing any new crystal solve, like new crystal, you don't know the structure. So
Mm.
how do you rule out those possibilities? Like there is no structural change of the cluster upon mechanical grinding, like whatever crushing or and all. So for that, do you need SCXRD data to confirmation, or are you enough reliable with that only micro ED?
No, I don't think. Maybe we can try this. Like if the crystals, if we can just try before grinding directly micro ED, then after grinding we do micro ED and we can relate the
But
studies
before
between them.
grinding, your crystals are much larger or like bigger size, so you can do.
In many of the cases we have seen, like some crystals we can just directly, just we will not grind them. Just we will crush them between the glass slides just to make them small and then do micro ED. And then after grinding, as you have suggested, we will do the mechanical force and then we can compare the results before and after.
Okay. And regarding your future direction, you are like telling that in a single grid, you have two type of polymorph. So these
Yes.
polymorphs are forming on your grid, or it is already formed in the crystallization vial?
It is already formed in the crystallization. It is not formed on the
But
grid.
they mentioned two type of crystallization will lead to two type of polymorphs, but you
Yes,
are getting
exactly.
purely
That's what the
one grid.
challenge we faced here. First challenge was I was not getting cubic polymorph at all. But recently, from the same, like where we are drop casting the, okay, I'll just explain. In AG29, there are two crystallization techniques. One is just dropcast cluster solution of DMF on the glass slide, and another is vapor diffusion. But in case of that direct dropcast of DMF, we are getting some powders. We didn't grind it or crushed it, we just put it on the TEM grid, directly loaded it, and we are getting both of them. So I believe that it is formed on the glass slide itself. Like we have
Like
not
it
used
is
both the
from
method.
the crystallization setup itself, not for your grid.
No,
It is not introducing any polymorphism.
no.
I think we are emphasizing here that we are getting two types of polymorph in a single
Polymorph
grid.
from the single grid
Rather, we
square.
can say that we can getting two type of polymorphs from a single type of crystallization set.
Yes, but my goal is because from single crystal XRD, if you want to know the like structure, you can mount at one crystal at a time. But here we can diffract these two crystals together, like on a single grid square. We don't need to mount it separately. So that's what my plan is right now.
Okay, got it. Thank you.
Any other question?
Yeah, I want
Yeah,
to.
Harshita,
Abhijesh, please go ahead.
just for this thing, the first paper is Utekol who diffracted 3D, right? 3D.
What, bhaiya?
The first paper by Utekol, not by this guy. The first 3D he has done.
Tamir Gonen group.
It's Utekol. or UTE KOLP. He's the one who did first this tomography.
Bhaiya, I cannot hear you properly.
I'm telling that this person, this lady you take call KOLB, she has done the first 3D. Like
Okay.
the electron tomography was done by her. Although the structure was solved by Tamir group, this the first 3D that was developed by this lady, Ute Kolb.
Okay.
So this.
Harshita, I have a small question on your slide 23. See, what you are seeing here, when you are grinding, the luminescence is changing to say yellow. And most probably it is the solvent effect, because when you are exposing it to DCM, right?
Yes.
DCM, then you are getting back the luminescence. So my question is, what do you think if you are grinding it, you are losing, like DCM has a very low boiling point and things. So DCM is vaporizing. And again, when DCM is coming into the crystal, it is again coming back to the crystalline phase. What?
Bhaiya, what I understood from here is when after grinding, emission is changing because there is a loss of the long range order. Suppose you have, for the formation of a crystal, we have many crystals, many clusters, which are arranged periodically. So when we are grinding them, this crystalline order or the long range order is getting disrupted. That's why its emission is changing and it become amorphous. But as we are exposing it to solvent, these solvent molecules are trying to reorganize those clusters and trying to reorganize those clusters, and that long range order is getting back. That is why emission is coming. So the role of solvent here is to reorganize and recrystallize the sample.
Harshita, you are
So
giving solvent vapor, right? Not the whole
Yes,
solvent.
correct. Solvent
That
vapor.
that won't be enough for reorganization to regain the long range order from a short range order crystal. It's like you are
Yes.
telling you are growing large crystal from a small crystal. That cannot be happening. That is not physically possible.
Yes.
So you have to tune the explanation in a different way. This is not possible.
Okay. Like I'm
Maybe,
saying that this solvent vapor help to reorganize them so that it can like it is coming back the same emission.
yeah, that's the same explanation you're giving before. Like short range order, like long range order is lost due to mechanochromic, like mechanic grinding, and you are converting the short range order to long range order by just adding vapors. That is not possible, right? Because you are telling what you physically mean is you are getting larger crystals by using smaller crystals and just adding vapor. It's like you are connecting two small crystal to become a large crystal. That's not possible, right?
and with the
There might be some
solvent
other
vapor.
explanation.
It is bigger crystal is going to smaller crystal. It is just order is broken. You can see amorphous nature. You can see.
That
That's
is
why
correct.
She is
That
not
is
talking
correct.
about that. She is talking about the extent of order. She is
Yes.
talking about the, let us say there is orientational disorder in this system. She is enhancing the translational order, reducing the orientational disorder, so which requires very little of change. For example, a molten crystal is recrystallized, or let us say from a glassy state is recrystallized. You are not getting you are getting great translational order by reducing the rotational disorder. So maybe the system is having certain kind of rotational disorder, as you can see in the carborane, there is a lot of rotations that are possible. Something is happening to the system.
Yes, sir.
Could
This
be.
explanation fits well.
Yes.
I have one question here. So since
Deepak, can I just continue that
yes,
question?
sure.
Yeah. So, Harshita, now my question is that DCM is playing a great role. So, if you are removing DCM in like not by grinding, by any other means, do you think that it will again go to amorphous and then it will again come to crystalline phase? If solvent is only the issue, say you are heating it, so it will lose crystallinity and the luminescence will change to yellow. And if you are again exposing it to solvent, it will come back. Do you really mean that?
Yeah, my objective was here to get the structure from the very tiny crystal. So I have not like optimized this. Like I have not tried without solvent or this, but we can try that. But I cannot comment right now without, like, I have not done that earlier.
No, no, this is a subsequent explanation what you are giving about the solvent. So I'm just tuning it a little, because in Arijit's paper, when he has given some heat, because DCM you need to give a little heat. So when he is giving heat, it is changing into yellow emission, but
Okay.
it is not coming back to green when exposed with the DCM vapors.
Means you are talking about thermochromic
Yes,
effect?
yes. I am just trying to correlate this mechanic properties with this thermochromic effect and
Thermochromic.
things like that. That's what I'm saying. If DCM is removed, say it is converting into yellow emission. But in the case of mechanochromic, this grinding, it is coming back to green. But in the
Yes.
case of thermochromic, it is not coming back. That means DCM is not the only factor.
Yes. DCM is not the only fact. But if you will heat it, the DCM that will also evaporate, no?
Yeah, and even heating it up to 100 degrees centigrade, it is not changing to yellow. It is going to yellow when it is heated up to say 120 or something. If I recall correctly from Arijit's paper. So that's what my thing is, that DCM is not the only factor which is playing a role here. There might be something else which is like
Is it
playing a
because
role in.
of the mechanical stress also which we are applying? Is it can depend on that as well?
Yes, it is possible. Indeed, it is possible. And secondly, one more thing is there. When we are heating it, we are not changing the size of the crystal. So the vapor, what
Yes.
we are exposing, a less surface area will be exposed to vapor, and might be it will take a very lot of time to come back to green. So that is also
Okay.
explanation. But I really feel that there is something else other than only solvent.
Yes, it could
Yes,
be.
that is what sir explained, right? Like when you put mechanical stress, there is orientational disorders which is changing the luminescence. But when you give the vapors, there is slight freedom for that crystal to again reorganize.
Yes. And Amog, the size also matters
Size
here,
also
because
matter.
this
Yes.
grinding thing, that size is very small. So you need less vapor exposure.
Yes.
Yeah.
Vivek, your like the question you ask actually can be explained. Like you mentioned that when you are heating, so DCMs are going out from the
Okay.
crystal, right? So that means gradually gradually it is now putting some disorder back. And that's why you need a certain temperature where all the probably DCMs are going out of the crystals, and then it's giving you the perfect disorder, which is giving back the luminescence.
Yeah, exactly.
Yeah. So I guess like it's, and I think we are like mingling two terms here. One is crystallinity and one is long range order and disorder. Both are different. You are
Yes.
talking about, Harshita, I think somewhere you told that it's like increasing the crystallinity and all. Actually, it's not increasing or
Not
decreasing.
increasing the crystallinity. Crystallinity is coming back.
Yeah, it's actually the long range order or the long range disorder is going and coming back. I think if you are giving the explanation is that manner, then all these confusions will evaporate up.
Then,
Okay,
Sujan,
okay.
the question will come, how much crystallinity is losing when you are grinding? So can you correlate the loss of the crystallinity and the shift in the emission from green to yellow? Can you do like instead of grinding for 20 minutes, grind for the 5 minutes, 10 minutes and 15 minutes to see the loss of the crystallinity and correlate with the
Exactly.
emission shift?
It's doable. Obviously, it's doable. What you are saying, so how much, like what we are doing under pressure. So that is hydrostatic, it is not hydrostatic. So depending on how much pressure you are putting, you are actually losing some kind of order in the crystal. And how, yeah, I think.
Yeah, you continue.
Yeah, so it depends on that how much ordering or disordering you are bringing into your crystal system, and depending on that, all things will get changed because it will affect your transition dipole moment. Depending on that, your like fluorescence wavelength will change, intensity will change. So definitely, it's like the effect has to be subtle. Like a subtle effect can also bring a lot of disorder.
Yeah. So continuing this, Harshita, since you are mentioning the role of the DCM, but you don't have the DCM in the crystal structure before
Yes.
before the grinding, even after the grinding. So how you can comment like DCM is playing the role to bring the luminescence in this one if you don't have the DCM in the crystal packing or even the void?
In the crystal structure, we didn't observe
So
DCM
if
molecule.
there is no DCM, then how you can comment like the DCM is evaporating? So that's why it's losing the emission from the green to yellow. And when you are exposing again, it's coming. So there is no role, a direct role of the DCM in the crystal packing. So you cannot comment from my understanding. Correlate this.
But
And
Bhaiya,
another,
when we
yes,
are exposing it to solvent DCM vapor, it is regaining its
yeah,
emission.
it's
So there
regaining.
must be a role of solvent.
But in
One
the
second,
crystal
just
structure, you
Deepak,
don't have.
I don't understand why this argument is going like when she's putting the pressure, it is going to short range order, which is amorphous, which has seen by Bhaiya's paper. Also, you can see in the electron diffraction. And it is well documented that if you exposure, it is more likely
Getting back
organically,
its emission.
more
Because
like
her objective is to understand this phenomena with the micro ED. So if some better explanations he can give. And what I am thinking like here is happening because if you remember the copper 4 with the iodine, the bond length between the copper is distort. In case when you are applying the pressure, mechanochromic, the bond length of the copper get increased. So it's luminescent change. But when you expose the bond length again restrained, the smaller. So that's how it gain the luminescence. Maybe that concept will be valid here. Also the symmetry breaking. Maybe when you are applying this pressure, is symmetry somehow distorting, which giving the different luminescence property. When you are exposing, if somehow is ordering, you can correlate with the ordering also. So symmetry may be playing the role here.
Deepak, symmetry is correlated to the intensity. Symmetry will not bring the luminescence wavelength down or up. Symmetry,
But
if symmetry is broken, you will just see the reflection will be in the intensity rather than in the wavelength.
okay, but the original copper for all this mechanochromic study started with the copper for iodine 4. So there they have mentioned the symmetry breaking in the copper 4 bond length distance. So you have to correlate with that, Sita. And another continuing this, I have another question like this. Can you mention like you have observed the toluene in the structure somewhere like other one? So before going to the micro ED, did you check with the same crystal before breaking it to the single crystal XRD?
No,
And
we have not checked.
because
With
this
the
is
same
maybe
crystal, we have not checked.
maybe because this is they have a squeeze while solving the single crystal XRD. They have not bothered about the solvent. We can squeeze it. So this is not something you will only observe in the micro ED. This can be
Okay.
observed with a high electron there is electron density residue in the single crystal XRD also by the X-ray. The only thing they have not modeled it. So that's the thing. They have squeezed this solvent. So there will be some void related to this.
And
From the
one,
single
But
yes,
if they
yes.
squeeze the solvent, will the that space group or unit cell parameter will not vary?
No, you are just squeezing. You are not considering that electron density related to the solvent. You are modeling. You are basically X-ray. You are propose a model. You are not considering the electron density related to the solvent. You are squeezing it, so it will not change.
And this MCBT, copper for MCBT, we have say crystallized it in different solvents. It is giving polymorphism. Okay. So in my question is this toluene, what is the source of this toluene? Because this crystal can be crystallized in a normal DCM. Say just you put it in DCM, it will crystallize. You will get the crystals. So what is the source of this toluene?
But yeah, the crystals which I got this, it was crystallized in a toluene, like DCM toluene mixture.
In that case, we have crystallized it in toluene, and Arijit has the data with the toluene also in the structure from single crystal X-ray diffraction. That's what
Okay.
Deepak was saying.
Yeah.
So
Okay.
we have this toluene as well as I don't remember exactly. There was one more solvent which was present. So we have some 5 to 7 structures. So that's what
Okay.
Deepak was saying. His point is valid that the same crystal you could have checked with the single crystal
Single crystal
extraction.
XRD.
And squeezing is like it is normal. Like we will squeeze all the solvents because we are not interested much in the solvent.
Yeah, got it.
So another question is related technicals. How you avoid the potential electron beam damage during this measurement, because this copper will be a little bit sensitive to the
We
electron.
have we use very low electron dose rate.
Okay.
Also, we use nanoprobe like just to select one particular area, like selected area electron diffraction.
Okay.
Okay, Harshita, for a diffraction pattern, like you can see here, I want to know three things from the diffraction pattern. First of all, like Soham also asked a bit, but I want to know what the radius signify. That's the first one. Second is how many rings are there? What does that signify? And the third one is what is the distance between two concentric rings are signifying?
Yeah, these concentric rings are drawn manually here, like in our software EPUD. Like if you will see it from the other software, it will just give you the diffraction spots. But to know the resolution in the software itself, they just draw it in such a way that we can get the idea of till how much angstrom these spots are going. Just to know the idea of resolution. Like there is no physical significance of these concentric rings.
I think they have physical significance. Please check. I think this is like how much penetration can happen, like inter
Also,
distance.
no,
Even I'm
Yeah.
telling
physical significance is about the diffraction spots and the distance between them.
No,
This, if
no.
you are asking
Sujan
only
is
about this
correct.
concentric
Yeah,
rings,
no. You please check. I
Sujan
think
is
yeah.
correct. It's
Yeah.
about how much resolution you have. So you have the beam spot from there. How good diffraction is there. The
I
better
that's
the
think
what
how
I'm
many
resolution,
telling.
the
planes
better
are
big the ring.
yeah,
like how
listen
many
to
planes?
listen. Am I
Yeah.
audible?
Yes.
Yeah. Go ahead,
Yeah,
Bijesh.
this is the thing, like two atoms. You are seeing the spots of the atoms, right? So that 2.49 Armstrong. So you are seeing the distance from the center to the
P
that
spacing.
particular atom. So yeah, this spacing what you were telling, or the from the center to that atom. This is what you are seeing from. If you can you pointed it, it's like a reciprocal lattice. She was
Yes.
talking about Ewald sphere and all these things.
Localist.
It's the reciprocal lattice which is you are seeing at the largest. If it is the largest distance, like 0.83 is the closest atom to the center atom. You understood? Ewald sphere works like the reciprocal lattice. Okay, the farthest distance spot is the nearest atom. So that's the resolution means 0.83 means you can see at least 0.83 if the atom is placed in the distance of 0.383 Armstrong, you can see that. Like it's very close. That's why we are seeing very closely, we are seeing the structure. That's why the paper science, they have published the hydrogen, location of hydrogen of paracetamol, they published in Science. So they can see the hydrogen atom in the diffraction.
With improved resolution.
The carbon and hydrogen and oxygen and hydrogen. These things are referring to that.
It's the same thing as diffraction resolution in any diffraction experiment. How closely can you see? And so you can measure that from your TEM images. Similarly, if those closely spaced lines can be resolved in here, lattice can be resolved, that is your resolution of your spectrometer or a microscope or whichever device that you have. Now you are based on that spots only you are creating the structure. So this is nothing but the resolution of the structure itself. Now, I have a question not on this, but on the previous some image, 23, I think. I want you to probably, can you expand B? Image B, expand that. Yeah, yes. Some more, can you expand? Yeah, so if you look at this, there are diffraction spots which are not having the same kind of intensities. See, between the two bright spots, there is a weak spot. And on the left side, you can see the intensities are decreasing. Somewhere there is this beam stopper, right? Beam stopper is there. Can you just point to that beam stopper? Yes, below that, there is one more line, a series of lines. Can you go to the right side of that line? So you see that if you go down, we are very close to that line. First circle, second circle. In between the first circle and second circle, there is a bright spot. Then there is a weak spot and the two weak spots. I want you to, and similarly, if you start looking at the bottom line, this is not something that you keep seeing as a continuous gradient is not there for the line for the spot intensity. What is the reason for this? Now, does it really mean in a particular crystal, you know, if you have a crystal, certain number of electrons are there in that lattice plane because of the atoms that are present. Now, if you have a few atoms less or a few clusters less in that lattice plane, the intensity that you have may be less. I do not know what is the correlation between these oscillating intensities along a particular Miller index. So now if you see left side of this, there are two bright spots and no other spots are seen which are bright. So some clarity is required here because these things will become important in the context of your later work or life. Okay.
Yes, I will look into it.
Yeah. Maybe it is important for Brijesh also for the kind of crystals that he is looking at. A few water layers are not there. You know, these are like you have a crystal and then a few atoms are missing or layers are missing and then another stuff. Then you have a step, atoms are missing. Now you are taking diffraction from this. Now it is not that, you know, you're microcrystals, right? You're not one crystal having almost the same density or same kind of atom density, I mean, across. In that 1 nanometer spot size itself, you have variation. Is that reflected here? Okay. That's all my question is.
Yes, I will check this.
So this is primarily because of the multiple diffraction from the same electrons, not. Yeah, so.
Multiple scattering?
Yes, multiple scattering from the same electrons. So so that's why we don't get like mirror image kind of a Friedel pairs. So Friedel's law is not really valid for electron diffraction. For at least for thicker samples. For very thin samples, it kind of holds onto. But for thicker samples, because of the multiple scattering, it doesn't really hold.
Very nice. Why is that certain intensities are lost, Anirban, on the right side of that two bright spots?
Still we'll have to figure out, but yeah.
Yeah, so anyway, these are certain details are there in these diffraction,
I have
the data that you have collected. Yeah.
Yeah, is it not because of the crystal orientation? But because in the single crystal XRD, when the crystal is rotating based on the detector distance as well as the orientation of the crystal, so some spot will be very intense. The same spot after the another orientation intensity will be low. So maybe similar kind of things is happening here when you are rotating the stage.
Or is it because of changing the planes?
Yeah, direction of the scattering. Because in the XRD, we notice the same spot in another frame, it will be intensity will be go down. Same thing will be happening here, I guess.
No, hello.
Yes.
Same thing will happen. I mean, same thing will happen and more.
Okay.
Then how can we distinguish that this effect versus multiple scattering? Or we can't?
That's why for electron diffraction, the
Dynamical refinement we have to do.
yes, I mean, dynamical effects become more
Because here there are
important.
chances of
So
multiple
that's why like Lucas brought dynamic refinement into it.
Yeah,
So
While
like
in
dynamic.
case of single crystal XRD, it only works with kinematical refinement.
but like your crystal is really, really thin, right? That is your USP, what you started with. That we
But
will.
here we have not used dynamical refinement. Here also we use kinematical. We are not considering the multiple
So,
scattering effect
so
here for this case.
if okay, then the next question is, if it is kinematic, then how are you talking about multiple scattering?
Because there is like, because of the presence of like,
No, for kinematic, you are at the first go itself, you are removing all the multiple scattering.
no,
Please
no,
make it short due to
kinematical
time
and dynamical
limitation.
comes later on.
Due to
First
time
we have to,
limitation, we have to conclude it also. So,
Yeah, okay, cool. I can catch Harshita after this. Yeah.
Don't
sir, do you
forget
have
to
any more
catch her,
questions?
okay?
Sorry, sir. What did you say?
Don't forget to catch her.
Yeah, absolutely.
Okay.
Sir, do you have any more questions?
I have no more questions. I really enjoyed her presentation as usual. She has been, well, very dynamic, and there are certain questions about certain details here because we are not really doing these. So it is more important to know about them, to know more. Now, of course, this points to several questions such as orientational disorder. What can you do about this orientation? How can you stop this orientational disorder? Now, you are doing this experiment at 77 Kelvin. I don't know whether it is 77 Kelvin.
good night.
But does it mean that then you need to have cool down even further? What's the kind of rotational order disorder that is seen in carbonates? At what point carborane stops? For example, methane, it will keep rotating even at 20 Kelvin. So you need to stop rotation, you need to go down to 10 Kelvin. But I don't expect carborane to be rotating to so low a temperature. But at the same time, this is one way. The other aspect is that if you don't want to stop, you don't want rotation, you functionalize it. This is how people do. C60, for example, it rotates, but you put a bromine there. Okay, it's a carbon-bromine bond. Yeah, then you stop rotation because it will hit on the next one to rotate. So it will stop. That is how crystal structures have been solved for such spherically symmetric systems.
Yes, sir.
Okay, so anyway, there are multiple questions or multiple pointers in this direction.
So those who have the question, please talk to her after the talk. So now I would like to conclude. Thank you so much, Harshita, for a very nice and interesting lecture. Definitely, micro-LED will be alternative techniques to the single crystal XRD X-ray. So we will meet again next month whenever slot will be available. So thank you so much everyone for joining.
Thank you.
Thank you so much. Have a good evening.
Thank you, Deepak, for coordinating this. Yeah, thank you.